ವೆಲ್ಕಮ್ ನನ್ನ ಮುದ್ದಿನ ಮಕ್ಕಳು, ಹೆಂಗಿದ್ದೀರಾ? Okay, I, Dr. Smita Narayan Karthik, is back with another beautiful chapter. And you know which chapter is that? Yes, it is the second PUC important chapter, and now we are going to talk about the next important chapter, that is human health and disease. It's a second PUC chapter. Andre nimge ella gottirbeku. That is what I expect, okay? Okay, let us start with the first question and the first subtopic of the chapter, that is common diseases in humans. Yes students, idu chapter nalli nimge gottirbodu tumba diseases ella discuss madiddivi, like malaria, like typhoid, and you know elephantiasis, filariasis, etc, etc. So let us throw some light on the important aspects of this chapter. Okay, so the first question here it goes: typhoid disease in human beings is caused by. The options are Plasmodium vivax, Trichophyton, Salmonella typhi, rhinoviruses. Now ನಿಮಗೆ ಸ್ವಲ್ಪ ಐಡಿಯಾ ಗೊತ್ತಿರಬಹುದು ಇದ್ರ ಬಗ್ಗೆ. Plasmodium vivax ಅಂದ್ರೆ which organism and what disease does it cause? Can you give me some hint? Yes, can you give me some hint students? Plasmodium vivax. Plasmodium name ಒಳಗೆ ನಿಮಗೆ ಮೀನಿಂಗ್ ಸಿಕ್ಕುತ್ತೆ. Plasmodium vivax. Yes, this is a one which actually causes the disease, that is malaria, right? So that is Plasmodium vivax which causes malaria. It's a protozoan, correct? It's a protozoan, kingdom Protista. You had studied in the first PUC, I think it was biological classification chapter. There you had studied kingdom Protista, and that is what we are going to take the first example, that was Plasmodium vivax is an organism which actually causes malaria. Okay, Trichophyton, it's basically a fungus which causes ringworm, athlete's foot, etc. So it's a fungal disease. It causes the fungal infection. It is very common ide, especially in between the finger idu irritation ಆಗ್ತದೆ. Then besides the groins, etc. On the scalp also, athlete's foot is one of the commonest example. Ringworm. Okay, so that is the group, that is a fungal disease group, and one such example here is the Trichophyton. Next is Salmonella typhi. Again, the name itself typhi indicates, yes, typhoid, and that is a bacteria, Salmonella typhi. And next one is rhinoviruses, group of viruses which causes common cold. Okay, now as you can make out from the name itself, you can indicate the answer. Typhoid disease is caused by the organism, that is Salmonella typhi, option C. Yes students, you know that a history of typhoid started with one lady, you know, Mary Mellon, who was actually a cook, and you know she was a main carrier of the typhoid fever, and she had spread it. She had once upon a time spreaded this disease to many because she was a cook in several places, and wherever she used to cook food, you know, especially the salads where we never use the heating concept, so that cases wherever houses she made the concept of cooking, she was a cook basically, and she had spreaded the disease. So that is how the discovery of typhoid came into existence. So the answer here, come back to the question, the answer here for the question here is Salmonella typhi. Okay, the next one, which one of the following diseases cannot be cured by antibiotics? Now antibiotics andre, why are we using in antibiotics? You must have visited any doctor, right? Antibiotics yake kodtare doctor? To treat the bacterial diseases, right? And now we want to jot down which one is the bacterial disease. Let us talk about that now. Plague, the options are plague, amoebiasis, leprosy, and whooping cough. Now if you look at the organism, plague is caused because of an organism, Yersinia pestis, right? Okay, amoebiasis is caused because of a protozoan, Entamoeba histolytica. Entamoeba histolytica. Leprosy, Mycobacterium leprae. And whooping cough by organism Bordetella pertussis. Okay, it's also called as pertussis. Whooping cough also called as pertussis. So if you see all the three out of the four options, plague, leprosy and whooping cough are actually caused by bacteria, and the amoebiasis is caused because of the entamoeba histolytica, which is a protozoan. Right? So which of the answer will be here correct? And that is going to be answer B, entamoeba histolytica. Okay, now we start with a very important life cycle of one major disease, and you know many people must have encountered the disease in their lifetime also. You know what are we going to talk about? Yes, I am talking about the next disease, that is malaria. Malaria gotta alla? How is it transmitted, students? Malaria? Mosquito bites, right? So you must be very scared about mosquito. The person who must have suffered or encountered malaria in the lifetime must be hating mosquitoes a lot, right? Okay, let us now talk roughly the life cycle of malaria, the malarial parasite. The malarial parasite is basically the plasmodium. Plasmodium. And there are different species of the plasmodium. Plasmodium is a genera, right? So you have Plasmodium falciparum, Plasmodium ovale, then Plasmodium malariae, etc. So now let us talk about the Plasmodium vivax, which causes the infection, that is malaria, a disease called as malaria. Okay, now this is, if you see here, the entire life cycle of the malarial parasite requires two hosts. How many hosts? Two hosts. One host you can make out from the image here. One host is the man, okay. Another host is the mosquito, and especially it is the female Anopheles mosquito. Female Anopheles only. Okay, so these are the two hosts which is required for completing the life cycle of this parasite called as Plasmodium vivax. Okay, since there are two hosts involved, this will be called as digenetic. What type of cycle it is? Digenetic parasite, because it requires two hosts to complete its life cycle. The two hosts are going to be, one is the man and the second host is the female Anopheles mosquito. So now let us talk about the life cycle in detail, and nimge gottirbodu idu. First of all, mosquito, okay, will come and bite a human, a man, okay. Mosquito actually bite ಮಾಡಿ ಆಮೇಲೆ ಏನು ಆಗ್ತದೆ ಅಂದರೆ, this mosquito, as I said, should be a female Anopheles mosquito, because female Anopheles mosquito will only be involved in the life cycle. So female Anopheles mosquito has got the type of the parasite, that is the Plasmodium vivax, in the form of sporozoites. In the form of what? The sporozoites. So once the female Anopheles mosquitoes comes for the blood meal, it will bites the human, and through the blood contact it will try to inject the sporozoites into man, right? Now what will happen to these sporozoites? These sporozoites will travel into the liver, and you know there are liver cells called as hepatocytes. Liver cells called as what? The hepatocytes. So this will, sporozoites will now attack the liver cells, that is hepatocytes, and will try to multiply in the hepatocytes, that is asexual reproduction. So once they multiply, they will form many such copies of the sporozoites, and now they are developed into the next stage called as merozoites. So we went to the next stage of the parasite. From sporozoite started the journey. Now we went to the merozoite, and slowly these merozoites starts rupturing the hepatocytes. So slowly the hepatocytes will burst and release the merozoites in the bloodstream. Now they will go and attack the RBCs. Correct? So you can make out here. I repeat once again, the mosquito will be biting a human, injecting the sporozoites form of the parasite. This sporozoite will actually go and attack the liver cells called as hepatocytes. In the liver, they will show multiplication by asexual method, and they develop into the next stage called as merozoites. Slowly these merozoites will come out, rupture from the hepatocytes, and slowly this comes in the bloodstream, and now they start hunting for the RBCs. Once they come into RBCs, remember student, that's a very important turning point of the entire life cycle. They start showing two type of multiplication. First of all, they will show asexual methods of multiplication, as well as sexual phase also. So let us understand what is the asexual methods of multiplication of the parasite. This merozoite slowly will develop into a ring stage, then the trophozoite, and then slowly a stage comes where it will try to rupture the RBCs. Once the RBCs are ruptured, actually you will find out that there is a compound called as hemozoin being released. You know what is this hemozoin going to do? The main symptoms of malarial fever. ನಿಮಗೆ ಗೊತ್ತಾ ಮಲೇರಿಯಾನಲ್ಲಿ ಇನ್ಫೆಕ್ಟ್ ಆದಮೇಲೆ ಏನು ಫೀವರ್ ಸಿಂಪ್ಟಮ್ಸ್ ಬರುತ್ತೆ? ಸಡನ್ ಚಿಲ್ಸ್, cold, correct, sudden dropping down and increase in the temperature. So that sudden shivers, all these symptoms are because of this compound, a chemical called as hemozoin. So hemozoin present in the blood will detect the presence of malaria. It is this stage where actually we confirm that there is a person exhibiting the symptom of malaria, right? Later what happens is that this life cycle continues in the asexual method, and there is one more sexual phase happening here. What is it going to be? Some of the merozoites then develop into the gametocytes, that is a male and the female gametes. So once this male and the female gametes are formed, they are now ready in the human, right? So slowly when the next female Anopheles mosquito comes and bites the human having the gametes, definitely this mosquito is going to take in the gametes. So what happens, ಏನಾಗುತ್ತೆ ಅಂದರೆ ಇದು ಬ್ಲಡ್ ಮಿಲ್ಗೋಸ್ಕರ ಇಲ್ಲಿ ಬರ್ತದೆ, ಕರೆಕ್ಟ್? ಆಮೇಲೆ ಇದು ಗೆಮೆಟೋಸೈಟ್ಸ್, ದಟ್ ಇಸ್ ಅ ಮೇಲ್ ಆ್ಯಂಡ್ ದ ಫೀಮೇಲ್ ಗೆಮೆಟೋಫೈಟ್ಸ್ ಹೋಗ್ತದೆ ಇದು ಮಸ್ಕಿಟೋಸ್ ಬಾಡಿ ಒಳಗೆ, ಕರೆಕ್ಟ್? ಬಾಡಿಯ ಒಳಗೆ ಹೋಗಿದ್ರೆ ಎಲ್ಲಿ ಹೋಗ್ತದೆ ಎಕ್ಸಾಕ್ಟ್ಲಿ ಇದು? ಇಟ್ ವಿಲ್ ಗೋ ಇನ್ ಟು ದ ಗಟ್, ದಟ್ ಈಸ್ ದ gut system, right? So once it reaches the gut, there will be the fusion to form oocyst, that is ookinete, that zygote, ookinete and the oocyst. So this is nothing but the sexual reproduction, that is a fusion of the gametes, correct? So once these gametes are fused, ಏನು ಫಾರ್ಮ್ ಆಗ್ತದೆ ಅಂದರೆ that is the oocyst. Slowly ಅದು oocyst ಡೆವಲಪ್ ಆಗ್ತದೆ ಸ್ಪೋರೋಸಾಯ್ಡ್ಸ್, and ಇದು ಸ್ಪೋರೋಸಾಯ್ಡ್ಸ್ ಆಮೇಲೆ ಮಸ್ಕಿಟೋಸ್ ಗಟ್ಟಿಂದ ಸ್ಲೋಲಿ ಟ್ರಾವೆಲ್ ಆಗುತ್ತೆ ಸಲೈವರಿ ಗ್ಲ್ಯಾಂಡ್ಸ್. ಸೊ ಸ್ಲೋಲಿ ಸಲೈವರಿ ಗ್ಲ್ಯಾಂಡ್ಸ್ ಒಳಗೆ ತುಂಬ ಸ್ಪೋರೋಸೈಟ್ಸ್ ಇರುತ್ತೆ. ಸೇಮ್ ಮಸ್ಕಿಟೋ ಬೇರೆ ಹ್ಯೂಮನ್ಗೆ ಬೈಟ್ ಮಾಡಿದ್ರೆ ಆಟೋಮ್ಯಾಟಿಕ್ಲಿ ಏನಾಗುತ್ತೆ, ದ ಲೈಫ್ ಸೈಕಲ್ ವಾಪಸ್ ಕಂಟಿನ್ಯೂ ಆಗುತ್ತೆ ಆ್ಯಂಡ್ ದ ಸ್ಪೋರೋಸೈಟ್ಸ್ ಆರ್ ಇಂಜೆಕ್ಟೆಡ್ ಇನ್ ಟು ದ ಹ್ಯೂಮನ್. ಸೊ ದಿಸ್ ಇಸ್ ಹೌ ಯು ಕ್ಯಾನ್ ಸಿ ದ್ಯಾಟ್ parasite requires two hosts to complete the life cycle, and that's the reason I said another MCQ can be asked. This parasite is going to be digenetic. So what do you mean by the digenetic? There are two hosts involved in the life cycle. One is the man and the second one is the female Anopheles mosquito. So this was a complete life cycle of the malarial parasite, and here as I quickly recap. So the mosquito bites and ejecting the sporozoites. These sporozoites attack the hepatocytes, that is the liver cells, and slowly they rupture the hepatocytes and then they develop into next stage called as merozoites. These merozoites go to the bloodstream and attacks the RBCs. So in the RBCs the two stage show ಆಗ್ತದೆ. ಏನಂದ್ರೆ first stage will be the asexual method of multiplication. Slowly ಆಮೇಲೆ ಅವರು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಮತ್ತು ಫಿಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಆಲ್ಸೋ ಫಾರ್ಮ್ ಮಾಡ್ತಾರೆ. ಅದು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಫಿಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಬ್ಲಡ್ನಲ್ಲಿ ಇದ್ದರೆ ಅದು ನೆಕ್ಸ್ಟ್ ಮಸ್ಕಿಟೋ ಬರ್ತದೆ ಫಾರ್ ದ ಬ್ಲಡ್ ಮೀಲ್. ಇಟ್ ವಿಲ್ ಗೆಟ್ ಇನ್ಕಾರ್ಪೊರೇಟೆಡ್ ಇನ್ ಟು ದ ಮಸ್ಕಿಟೋ. ಮೀನ್ವೈಲ್ ಇಲ್ಲಿ ಒಂದು ಕಂಪೌಂಡ್ ಸಿಕ್ರೇಷನ್ ಆಗ್ತದೆ. ವಾಟ್ ವಾಸ್ ದಟ್ ಕಂಪೌಂಡ್ ಸ್ಟೂಡೆಂಟ್ಸ್? ಹಿಮೋಜೋಯಿನ್. ಅದಕ್ಕೆ ಹೇಳ್ತಾರೆ ಅದು ಹಿಮೋಜೋಯಿನ್ ಸಬ್ಸ್ಟೆನ್ಸ್ ಏನು ಮಾಡ್ತದೆ ಅಂತ ಸಿಂಪ್ಟಮ್ಸ್ ಕ್ರಿಯೇಟ್ ಆಗ್ತದೆ ಬಾಡಿಯಲ್ಲಿ. ದ್ಯಾಟ್ ಈಸ್ ಅ ಇಂಡಿಕೇಟರ್ ಆಫ್ ಮಲೇರಿಯಲ್ ಫೀವರ್. ಸೊ ಆಮೇಲೆ ಅದು ಮಸ್ಕಿಟೋ ಒಳಗೆ ಗಟ್ನಲ್ಲಿ ಅದು ಪ್ಯಾರಾಸೈಟ್ಸ್ ಫಾರ್ಮ್ ಇದೆ ಇನ್ ದ ಗ್ಯಾಮೆಟೋಸೈಟ್ಸ್ ಫಾರ್ಮ್. ಸ್ಲೋಲಿ ಆಮೇಲೆ ಅದು ಸಲೈವರಿ ಗ್ಲ್ಯಾಂಡ್ಸ್ನಲ್ಲಿ ಹೋಗುತ್ತೆ ಆ್ಯಂಡ್ ಇಟ್ ವಿಲ್ ಗೋ ಆ್ಯಂಡ್ ಬೈಟ್ ಅ ನ್ಯೂ ಹ್ಯೂಮನ್. ಸೊ ದಿಸ್ ಈಸ್ ಹೌ ಇಟ್ ಕಂಪ್ಲೀಟ್ಸ್ ದ ಎಂಟೈರ್ ಲೈಫ್ cycle. So now let us talk about some MCQs related to this concept. Okay, so here we go. So the question for you all is a toxic substance, okay, responsible for chills and high fever. So look at the symptoms students here. ನಾನು just ಹೇಳಿದೆ ಅಲ್ಲ, ಮಲೇರಿಯಾ ಫೀವರ್ನಲ್ಲಿ ಒಂದು ಸಿಂಪ್ಟಮ್ ಕಾಮನೆಸ್ಟ್ ಬರ್ತದೆ and that is a symptom here. So what is the symptom here? Sudden fever and high chills. Okay, chills and high fever recurring every 3 to 4 days in malaria, and that is because of what compound? Yes, any guesses? The options are interferon, hemozoin, herodin, colostrum. Now let us try to understand quickly what are the four options. Interferon students means they are antiviral proteins. Antiviral protein andre ಏನು? See, it is a very important chemical. When we are going to talk about immunity, we'll be throwing again light on that. You know, there are viruses, okay, which will infect the normal cells. So once the virus are infected by some pathogen or the virus, okay, this normal cell will produce certain chemical in response to this pathogen, and that is called as interferon. Right? So this interferon, the chemical which is produced right now, will go and protect the neighboring healthy cells. So these neighboring healthy cells will be protected because of the interferon, and the virus again will not come and attack the neighboring cells. So that's why it's called as antiviral protein, which again acts as a defense mechanism protecting the neighboring cells. Right? Let us go to the next point, hemozoin. As we studied right now, hemozoin is a chemical substance produced by the rupturing of the RBCs during the life cycle, that is asexual cycle of the parasite Plasmodium vivax. Next is a hirudin. Hirudin is basically an anticoagulant. And the last option is the colostrum. Colostrum is the yellowish fluid present in the milk, that is the lactating milk, and yes, it is found to be rich in antibiotic. So antibody, rich in antibody. That will be again talking about this. So this colostrum is a yellowish fluid present in the first milk of the lactating mother, and yes, it serves a very important function of defense mechanism. So out of the 4 options, which are the correct answer here? And yes, you know it very well. The answer here is hemozoin. Next question. Which of the following set of diseases are caused by bacteria? Options are tetanus and mumps, herpes and influenza, typhoid and smallpox, cholera and tetanus. Now students, let us make it more simple. Try to strike out the diseases which are actually caused by the viruses. And here they are. First is the mumps virus, causes mumps. Then the herpes, influenza, and the smallpox. So these are the viral diseases. Now which are the ones which are left out? Tetanus, typhoid, cholera and tetanus, right? So if you see in the above 3 cases, they are the bacterial diseases are clubbed with the viral diseases. So of course they are not the right answers. And now let us see the right answer is going to be the last option left out. If you see, cholera and tetanus are the bacterial diseases. Cholera is caused by an organism, that is Vibrio cholerae, and tetanus again a disease caused by Clostridium tetani. So the answer here would be option D, that is cholera and tetanus. Now what we are going to talk here is about the very beautiful and the core of the chapter, that is the immune system. And here is a diagrammatic presentation of what does the immune system do, right? Yes student, ನಿಮಗೆ ಗೊತ್ತಾ ಇದು ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ ಎಷ್ಟು ಕಾಂಪ್ಲಿಕೇಟೆಡ್ ಇದೆ and so beautifully designed ಇದೆ? Let us now talk about that. Why do we require the immune system? Yes students, if you see daily ನಾವು ಹೊರಗೆ ಹೋಗ್ತೀವಿ, ಕರೆಕ್ಟ್ ಅಲ್ಲ? ಸೊ ಡೈಲಿ ನಾವು ನಮ್ಮ ಸರೌಂಡಿಂಗ್ಸ್ಗೆ ತುಂಬ ಜರ್ಮ್ಸ್, ಪ್ಯಾಥೋಜನ್ಸ್ ಎಲ್ಲ ಇರುತ್ತೆ. Definitely there are chances that we all consume or inhale the pathogens or the germs surrounding us. So don't you think so? If we would have ingested all and they would have all caused the infection, we would have not survived so far, right? Yeah, but don't you think so? Because of the gracefulness of the immune system which is present in our body, we are protected every now and then. Yes students, ಇದು ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ ಎಷ್ಟು ಸ್ಟ್ರಾಂಗ್ ಇದೆ ಅಂದರೆ ಸಾಧಾರಣಗೆ ಅದು ಯಾವುದೋ ಆರ್ಗ್ಯಾನಿಸಮ್ಗೆ ಇನ್ಫೆಕ್ಷನ್ ಮಾಡಲಿಕ್ಕೆ ಕೊಡೋದಿಲ್ಲ ನನ್ನ ಬಾಡಿಗೆ. ಅಂದರೆ ಅಷ್ಟು ನಮಗೆ ಪ್ರೊಟೆಕ್ಟ್ ಮಾಡ್ತಾ ಇದೆ ಇದು ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್. ಸೊ ಇದ್ರ ಬಗ್ಗೆ ತುಂಬ ಜಾಸ್ತಿ ನಾವು ಸ್ಟಡಿ ಮಾಡ್ಬೋದು, ಕರೆಕ್ಟ್ ಅಲ್ಲ? ಸೊ ಲೆಟ್ ಅಸ್ ನಾವ್ ಲರ್ನ್ ಮೋರ್ ಆ್ಯಂಡ್ ಮೋರ್ ಅಬೌಟ್ ದ ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್. ಆ್ಯಂಡ್ ಹಿಯರ್ ವಿ ಗೋ. ಸೊ ಎಂಟೈರ್ ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ಗೆ 2 ಪಾರ್ಟ್ ಇದೆ, ದ ಮೇನ್ ಪಾರ್ಟ್. ಸೊ ದ 2 ಮೇನ್ ಟೈಪ್ಸ್ ಆಫ್ ದ ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ ಆರ್ innate immunity and acquired immunity. I repeat once again, first type of the immune system part ಏನು innate immunity, ಮತ್ತೊಂದು immunity ಇದೆ acquired immunity. Main difference ಏನು innate and acquired? Innate ಅಂದ್ರೆ inborn, from the birth ಅದು ಇರುತ್ತೆ, correct? So innate ಅಂದ್ರೆ inborn, from the birth, right? Inborn. And acquired, grammatically acquired means you acquire it, you possess it later, you develop it later during your journey of your lifetime, correct? So acquired meaning slowly, gradually we try to develop that immunity, hence it is called as what? The acquired immunity. So that's the first difference. Innate andre inborn, from the birth irutte. Acquired from birth irudilla, later it is developed when we are exposed to different germs surrounding us, right? What is the second important difference? Innate actually non-specific irutte. ನಾನ್ ಸ್ಪೆಸಿಫಿಕ್ ಅಂದರೆ ಪಾಪ ಇನ್ನೆಟ್ಗೆ ಜಾಸ್ತಿ ತಲೆ ಇರೋದಿಲ್ಲ, ಅಂದರೆ ಅವರಿಗೆ ಸೆಲ್ಫ್ ಸೆಲ್ಸ್ ನಾನ್ ಸೆಲ್ಫ್ ಸೆಲ್ಸ್ ಏನು ಗೊತ್ತಿಲ್ಲ. ಅವ್ರದ್ದು ಡ್ಯೂಟಿ ಏನಂದರೆ ಓನ್ಲಿ ಪ್ರೊಟೆಕ್ಷನ್ ಆ್ಯಸ್ ಅ ಬ್ಯಾರಿಯರ್ ಅಂದರೆ ಯಾರಿಗೆ ಒಳಗೆ ಎಂಟ್ರಿ ಕೊಡೋದಿಲ್ಲ ಅಷ್ಟೇ. ಸೊ ಇಟ್ ವಿಲ್ ನಾಟ್ ಅಲೌ ದ ಎಂಟ್ರಿ ಆಫ್ ದ ಜರ್ಮ್ಸ್ ಸೋ ಈಸಿಲಿ. So they act as a barrier. You know, like a kingdom, a big palace will have a boundary. So like the same way, the innate will majorly play the role acting like a barrier. ಸಾಧಾರಣಕ್ಕೆ ಅದು ಆರ್ಗನಿಸಮ್ಸ್ಗೆ ಎಂಟ್ರಿ ಮಾಡಲಿಕ್ಕೆ ಕೊಡೋದಿಲ್ಲ. That's why we can say it is kind of a first line of defense, but then yes, it will act as a barrier. Moreover, it is not that specific. Yes, but what about this one? This is so complicated and so smart and well planned that they are very specific. ಅವರಿಗೆ ಸರಿಯಾಗಿ ಗೊತ್ತಿರ್ತದೆ ಯಾವುದು ನಮ್ದು ಸೆಲ್ಸ್ ಅಂದರೆ ಸೆಲ್ಫ್ ಸೆಲ್ಸ್, ಯಾವ ಸೆಲ್ಸ್ ಔಟ್ಸೈಡ್ ಇಂದ ಬರುತ್ತೆ ಅಂದರೆ ಫಾರಿನ್ ಸೆಲ್ಸ್ ಆ್ಯಂಡ್ ಸೆಲ್ಫ್ ಸೆಲ್ಸ್ ಡಿಸ್ಕ್ರಿಮಿನೇಷನ್ ಇದು acquired immunity ಬರುತ್ತೆ. ಗೊತ್ತಾಯ್ತಾ? That's why we use a word. They are specific defense mechanism and they are non-specific defense mechanism. Right? So that's a very important attribute of innate as well as acquired. So this is a brief outline of what are the different mechanism or the cells involved in the innate and the acquired immunity. So the first, we can divide them in 3 levels. The first line of defense, the second line of defense, and the third line of defense. Why do you think there is first line, second line and third line? See, first time the pathogen nam surrounding ide, correct? Our body will try to minimize or prevent the entry. ಯಾರು ಗ್ಯಾರಂಟಿ ಕೊಡ್ಲಿಕ್ಕೆ ಆಗೋದಿಲ್ಲ. ಸಮ್ಟೈಮ್ಸ್ ಇದು ಬ್ಯಾರಿಯರ್, ಸ್ಕಿನ್ ಬ್ಯಾರಿಯರ್ ಮೋಸ್ಟ್ಲಿ ಫೇಲ್ ಆಗ್ಬೋದು, ರೈಟ್? ಆರ್ ಇಸ್ ನಮ್ದು ನೋಸ್ ಸಮ್ ಆರ್ಗನಿಸಮ್ಸ್ಗೆ ಎಂಟ್ರಿ ಕೊಡ್ಬೋದು. ದೆರ್ ಆರ್ ಸಮ್ ಆರ್ಗನಿಸಮ್ಸ್ ವಿಚ್ ಆರ್ ವೆರಿ ಸ್ಮಾರ್ಟ್, ದೇ ನೋ ಹೌ ಟು ಎಸ್ಕೇಪ್ ದ ಬ್ಯಾರಿಯರ್ಸ್ ಆಲ್ಸೋ. ಸೊ ದಟ್ ಟೈಮ್ this comes into picture. Slowly this and then this. So we have divided the defense into 3 categories. So first line of defense is going to be just entry level will be restricted at the first time. Yes, I mean the first level of entry, okay, will be prevented. If at all the organism is very smart enough, it will escape this, and later when it enters the body, it will be encountered by the second line. If the organism escapes even the second line of defense, then there is third important type which is actually called as a third line of defense. Third line of defense is very specific, very strict. In case if a germ is escaping these 2 line of defense, third line will not allow the organism to establish in the body so easily. So that's why all the God has gifted us with all the 3 lines of defense mechanism. So what are the different cells, tissues here? If you see, the first line of defense will include the skin, right? Then the mucus, right? And then the secretions of the skin and the mucous membrane. I give you quickly some examples. Skin andre nodi, idu skin ge outermost layer irtade, keratinocytes, correct? So again, it is itself a barrier. Easily adu bacteria ge entry madlikke kodilla. That's why it's a first line of defense. And there is something on mucous membrane. Mucus, a sticky substance, yes, which is present in the, the nose having the mucus, the sticky substance. Don't you think even that is really playing a very important role in trapping the germs? So when you inhale the air, germs irutte side nalli, surrounding nalli. So what happens andre, idu mucous membrane ಏನು ಮಾಡುತ್ತೆ ಅಂದ್ರೆ, ಅದು mucus sticky substance ಅದು germs ಗೆ trap ಮಾಡ್ತದೆ and ಅದಕ್ಕೆ ಒಳಗೆ ಹೋಗಲಿಕ್ಕೆ ಬಿಡುದಿಲ್ಲ. That is what the creativity of God. You should really thank God for creating small structures and tissues and fluids being made in this manner, right? So that is how mucous membranes and secretions of the skin. Yes, secretion of the skin andre, oily secretions being done by the skin, that is sebum, having the pH level. Yeah, so sebum also acts like a secretions, which again a first line of defense. Now come to the second line of defense. So here we go. You have something called as a phagocytic cells. Phagocytic. Phagocytic cell matlab eating up. So there are special group of cells which will try to eat up the pathogen. Andre special cells irutte, like amoeba eating up the food, same way, the concept same, but here there are different cells which will try to eat the pathogen and digest it and try to destroy it. So they are called as phagocytic cell, which will play this role by the process called as phagocytosis. Students, mark this word, it's a very important word. Phagocytosis, it's a process of ingestion of the pathogen and then digesting it, eating it. So there are some specialized cells which do this role called phagocytosis. One is a macrophage, we'll be talking here. So macrophages will come here also and will play a role there also. You have second group of cells called as NK cells, which is nothing but the natural killer cell. And they have some chemicals which will try to destroy the pathogen and the inflammatory response. Next comes the acquired defense mechanism, that is the third line of defense, and these are the main cells. The lymphocytes, the lymphocytes ali ಎರಡು type irutte, B cells and the T cells. B cells and the T cells, or else B lymphocytes and the T lymphocytes, are the two main type of cells which takes care of the beautiful, well-planned, acquired immunity. Yes, students? So B cells will be and T cells both are going to show their own type of the immune responses. Little different they are. Yes, but they will take care of the third line of defense. Next comes the antibodies, again immunoglobulins, which are secreted by the B cells, which tries to destroy the pathogen, that is the antigen. And then macrophages. So similarly, macrophages here will also play a role in trying to eat up the pathogen and presenting it on the surface. That's why they are also called as antigen presenting cells. Antigen presenting cells andre there are cells like the macrophages and other group of cells which will eat up the pathogen. So our pathogen ge ingest ಮಾಡ್ತಾರೆ, ಆಮೇಲೆ ಅದು pathogen ge our surface ನಲ್ಲಿ project ಮಾಡ್ತಾರೆ. And this pathogen which is projected on the surface of the macrophage, this cell will be called as antigen presenting cell. Antigen andre namdu pathogen. Okay, it's a very proper term used here in immunology, antigen. ಆಮೇಲೆ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಇದು antigen surface ನಲ್ಲಿ ಇರುತ್ತೆ ಅಲ್ಲ, ಅದು macrophage surface ನಲ್ಲಿ, ಅದು B cell and T cell ಆಮೇಲೆ ಅವರಿಗೆ completely destroy ಮಾಡ್ತಾರೆ. So that is how it's a beautiful mechanism, an array of cells, it's a complete system which works together and many cells are involved together for the functioning here. So this was a brief outline of what is our entire immune system comprising of. So I can give you briefly, there are two types, one is the innate immunity and another is the acquired immunity. Let us now move on to the MCQs based on this, and I have homework for you, okay? So the homework ಮಾಡಬೇಕು, okay? I give you some hint on that. Which of the following barriers are considered to be the first line of our body? First line of defense. Okay. Options are physical and physiological barriers, physiological and cellular barriers, cellular and cytokine barriers, physical and cellular barriers. So there are four type of barriers basically in the first line, okay? So physical and physiological barrier, and if you see cellular and cytokine barrier. Cellular and cytokine barriers are mainly dealing with the chemicals being provided, and yes, in case of cytokine I'm telling, and cellular will deal with moreover the types of cells which are involved, like the macrophages. So you are going to get a hint, that is physical and physiological barriers are considered to be the first line, as I said, physiological barrier, and cellular cytokine form the second line. So these are going to be the second line of barrier. Okay, so your answer is going to be which one? Yes, you're very right. The answer is going to be option A. Okay, now we move on to the next, again a neat question being repeated. Which one of the following acts as a physiological barrier? Physiological, okay, physiological barrier to the entry of organisms, microorganisms in the body. So the options are tears, monocytes, skin, epithelial of urogenital tract. Okay, now if you see the physiological barrier, matlab it's going to try to kill the cells, right? Or the cell wall, and that is the tears. Tears actually is found to have the enzyme that is lysozyme. Yes, so these lysozymes are basically having the ability to kill or to destroy the cell wall of the organism, which is actually very important aspect of the bacteria. So the steers which will be present having the lysozyme will try to kill the cell wall, and yes, according to you the answer would be what? Having the physiological role, that is going to be the tears, option A. Okay. Next one, which of the following are not the phagocytes of innate immunity? I come to the point, phagocytes. If you remember, these are special type of barriers, that is a cellular barrier, which again help in the immunity. Now, what was the purpose of phagocyte cells? Yes, I said there was one concept called as phagocytosis, right? What was this phagocytosis? Phagocytosis is eating up of the organism, eating of pathogen by a cell called phagocyte, by phagocyte cell, correct? So eating off of the pathogen by a phagocyte cell, that process of ingesting it and then digesting it, ಅದು process ಗೆ ನಾವು ಏನು ಹೇಳ್ತೀವಿ? Phagocytosis, right? Okay, so phagocytosis, the cells which are going to show this property will be called as phagocytes. So far you have studied neutrophils, macrophages and monophytes. All the three options will be showing phagocytosis, but not the RBCs, right? So the option here is phagocytes basically is the process which will be shown by especially the phagocyte cells. Okay, like the monocytes, the macrophages, etc. Right? So the option here, as I said, would be what? The RBCs, which actually does not show this property. Correct? Look at that, the very important aspect. Macrophages are also modified monocytes, which are present in the tissues. Okay. Answered C. Now, antibody consists of. So here we need to know briefly the structure of the antibody molecule. Yes, students. Now, which cells will produce this antibody? As I said, you know there are two type of cells, the B cells and the T cells, right? So these were the two type of lymphocytes which were able to show the third line of the defense mechanism, right? So amongst the B and the T cells, it is the B cells which are going to produce the special protein called as antibody. Special proteins, or you can say immunoglobulins, called as the antibody, and it is this antibody will play a very defensive role, which will try to destroy the antigen. So antigen is nothing but the opposite of antibody, meaning antigen is nothing but the pathogen, okay, which will be entering our body, and yes, this will be destroyed easily by the antibody. So that's a word, that is antibody. Anti means against the antigen. So antibody structure we are going to learn now quickly. And yes, what about the T cells then? Yes, students, T cell as such will not produce antibody. Correct? Not produce antibody. They are not known to produce antibody. Yeah, but then they have their own mechanism of destroying the pathogen. You know, there are various type of cells which are involved in the T cell immunity, right? Which is called as a cell mediated immunity. So this is what the basic difference in the B cells and the T cells. Let us now move on to the structure of the antibody. Okay, so if you see, antibody is a typical Y-shaped structure. It's a Y-shaped structure. It is basically a protein. So it is made up of protein, right? It's also called as immunoglobulin. Also called as immunoglobulin. Immuno because it refers to immunity, globulin proteins, right? Now, what is this comprising of? The Y-shaped antibody will have two type of chains. So this is one type of chain, right? And this is a second type of chain. So these are the four chains which are present. These are the four polypeptides. So you find four polypeptides. Correct? 4 polypeptides. So these are going to be the outside ones are going to be the light chains, which are how many in number? 1 and 2. So 2 light chains, 2 light polypeptides, correct? And these are the 2 heavy polypeptides. Right? So you get the complete antibody having 4 polypeptide chains. Now, what is present here? You know, this end, since it's a protein, you know the protein has got one amino terminus and one carboxylic acid terminus. Amino and carboxylic terminus. Correct, no? Always, this you remember in biomolecule of first PUC we had studied, a protein has got two ends. One is amino terminus and one is the carboxylic terminus, right? COH terminus. So similarly, you will find that the amino terminus will be present at this end, which is supposed to be binding or attaching with the antigen. That's why it is called as the antigen binding site. So you can imagine if there is an antigen of this specific shape, it will be binding up in this way. So that is how it is going to form a complex, and then later it will destroy the antigen. So this site is very important. It is called as what? The antigen binding site. And the another one will be called as the part which is actually attached with the B cell. Yes, and that is going to be the carboxylic end. So this all the four polypeptides are connected with something called as what? The disulfide linkages. So this is what the disulfide linkages which is present here, which connects the two heavy chains, and in turn also these heavy with the light chain. So this was the structure of the entire antibody, and now we look on to the question of an antibody. An antibody comprises of two light peptide chains and two heavy peptide chains, two light peptide chains and one heavy peptide chain, one light peptide and one heavy peptide, and one light and two heavy peptides. What is the answer here? Can you tell me? I can hear you thinking yes, and the answer here is two light or two heavy. It is going to be two peptide chains and two heavy chains. Two light and two heavy chains. Correct? Okay. And this is for homework, students. So you are going to come with the answer here. Identify the marking A, B, C and D in the figure given below and select the correct option. So these are the options, and you will be thinking about the answer and coming back with your next section.